Hello, everyone. We're Group 11, and today we are giving the value proposition of our intra-blood-brain barrier glutamate monitor and anti-epileptic drug delivery system. So we came into this project intrigued with the very enigmatic biomedical challenge of crossing the blood-brain barrier. And during our research, we found that the diagnosis and treatment of epilepsy fits that bill 100%. Epilepsy is characterized by seizures, and this abbreviated list of causes shows that its epidemiology is varied, to say the least. To compound these problems, somewhere between 50 and 65 million people worldwide suffer from epilepsy, and that's about 2.2 to 3.5 million people in the United States. There are numerous social and economic hardships associated with this disease, any of which can worsen the others. But it's particularly challenging, both for patients and clinicians, in that about a third of epileptics don't respond to typical drug therapies. We hope to help this population by focusing on the neurotransmitter called glutamate.
Studies have shown that epilepsy is correlated with an increase in glutamate levels, which leads to a decrease of tight junctions. The decrease means that serum proteins can be released, leading to what is called a leaky blood-brain barrier. This leaky barrier leads to serum proteins coating the surfaces of drugs trying to reach the brain, leading to anti-epileptic drug resistance. To address this problem, we need to target GABA production, an inhibitory neurotransmitter that aids in the prevention of seizures. An increased glutamate level could trigger the release of emergency medication, clonazepam, which would increase the effectiveness of GABA. As shown in the figure, our device would be an implantable brain sensor that would detect the concentration of glutamate. The sensor would trigger the clonazepam release when the maximum glutamate threshold is detected, which would be released by the subcutaneous drug delivery system located on the patient's arm. The device will be quantifiable by detecting the specific concentration of glutamate in the brain. The device would be programmed to detect when the glutamate reaches the threshold, which will be translated to the amount of clonazepam that would be released. Finally, thinking about our stakeholders, the end users of our device would be individuals with severe, uncontrollable, and irregular epilepsy. Ideally, the insurance company would pay for the implant, and our client would be the medical device company that would fund the development of our device.
This device would have a few different value aspects. First is that it provides the patient with the needed drug to suppress seizures. A glutamate increase is not something the individual can detect on their own, so having a sensor that can detect those levels is essential to delivering the needed medication prior to the onset of a seizure. This improves quality of life for the patient by making it something the patient doesn't have to think about. Much like an insulin pump, the device would detect the problem in the body and send a response to help correct it. Another valuable aspect is the data that it provides for future research. By recording glutamate levels over time, this can allow for further correlations to be made regarding the onset of seizures and daily activities and how glutamate causes seizures. There are a few different costs associated with this device, the first being the cost of research and development for the device itself. Ideally, this would be taken on by a medical device company that can produce the device after approval. Current sophisticated sensors that are implanted in other areas of the body can cost less than $20 to produce using batch fabrication techniques. This would ideally be applied as well to our project, making it low cost. Once the device is on the market, it would likely be covered by insurance for the patient. This would include both the cost of the device as well as surgery itself. A downside to this device is that intracranial surgery is incredibly high risk for the patient as well as expensive.
To close, we are going to discuss some of the products in the market or in development that may serve as competition for our device. There are several products on the market today for treatment of drug-resistant epilepsy. Surgically removing the area of the brain where the seizures are focused, known as focal resection, is possible when the area affected is non-critical. For others, nerve stimulation devices are utilized. The three most common of these devices include vagus nerve stimulation, responsive neurostimulation, and deep brain stimulation, which all act as sort of pacemaker systems for the heart in varying ways. Beyond actual epilepsy treatments, glutamate receptors are a market of competition for our design. One product currently in development is a 3D printed biosensor. This device is meant to be implanted in the spinal cord to read for extracellular glutamate concentrations. The drawbacks of this design include, first, that implementation into the spinal cord limits real-time reading of the brain where seizures originate, and two, this device only collects information on glutamate concentration while doing nothing to actually improve them. Thank you for listening, and we look forward to any feedback.